So now we'll pick up where we left off. So now we're up to step 4, which is where we have alpha-ketoglutarate. Oh, one thing we did talk about, alpha-ketoglutarate is the alpha-keto acid of what amino acid? Glutamate. Okay? Once again, all you do for those alpha-keto acids is if you transform the ketone to the amino group, which we'll see aminotransferases, that's what they do, then that is glutamate. Just like oxaloacetate, OAA, is the alpha-keto acid to aspartate, and pyruvate is the alpha-keto acid of what amino acid? Alanine. So that's why those are probably the 3 things that I would expect you to know their structures, just because you already know the structures of their amino acids. Okay, so when you take NAD+, you get NADH. It does utilize coenzyme A, and this is where we get succinyl-CoA. I will just tell you right now, this one is also, even though it's reversible, it is a checkpoint. It is one of the ways that they keep the cycle under control. Okay. So, what class or classes of enzymes can this be? Or should I let me do? Does it look an awful lot like anything that we've seen thus far?
Dehydrogenase.
It's a dehydrogenase. Yes. And why is it a dehydrogenase? Okay, NAD going to NADH. Yes. Does it look like anything that we have seen so far this semester? Look at what all it requires. It looks like that complex and left complex was pyruvate dehydrogenase complex, and this is very analogous to that. So now put the pieces together, so what would you call this one? What enzyme is this?
Alpha-ketoglutarate dehydrogenase complex.
Exactly. The alpha-ketoglutarate dehydrogenase complex. It's very, very, very similar to the pyruvate dehydrogenase complex, but it's a pyruvate. We're working with alpha-ketoglutarate, and we get succinyl-CoA instead of acetyl-CoA. So that's the big difference. And I would never ask you how many kilojoules per mole, all that kind of stuff, but it is. It requires the same, TPP, lipoic acid, FAD, acetyl-CoA, and NAD. Same setup as before. Okay. Are there any questions so far? Anything? Okay, remember, and so we've actually gone over the checkpoints already. Not that step 0, pyruvate dehydrogenase complex, is kind of like a gateway. Then we had steps 1, 3, and 4, citrate synthase, isocitrate dehydrogenase, let's try it because I don't have the numbers memorized together, and alpha-ketoglutarate dehydrogenase complex. So now we have succinyl-CoA, which remember succinyl-CoA is actually one of the things that can feed back and inhibit citrate synthase, or step 1. Okay. I give you a moment to look this over. We're going from succinyl-CoA to succinate. What's one observation that you can make about this? And there are lots of them, but what's one?
It's one-sided.
It is one-sided. It's essentially irreversible.
It utilizes inorganic phosphate.
It uses inorganic phosphate. What else does it utilize? GDP. Okay. So because we make, what do we make from that? GDP. GTP. Okay. I think this is the first time that we've seen GTP made directly on something. Okay. So yes. Okay. And we get our coenzyme A back, so that way coenzyme A can be shuttled to either go back into the cycle here or go on to build fats if you wanted to build fats. Usually if this is going through, you wouldn't be wanting to build fats because you do this when you need energy. Okay, but now the $25 million question. Not really. $25 million hypothetical dollar question. What class or classes of enzymes could this be, knowing what we know? What's one of the possibilities? It could be kinase, if we were phosphorylating something. What else could it be? Along the same line. A what? I didn't hear you.
I just don't think I'm right.
Oh. What are some of the other possibilities? A what?
Oh, it has inorganic phosphate.
It's inorganic phosphate. So yeah, maybe a phosphorylase. This one, I mean, it does utilize GTP, and so we could say a kinase. What else could we call it?
Transfers.
Okay, and what would it be transferring?
Coax,
The acetyl-CoA, well, not acetyl-CoA, but coenzyme A. If it's going backwards, you're getting. Now, if we start putting those together, you're getting it really, really close. This, by the way, is actually named for the. It doesn't make sense to me. It's named for the reverse, which doesn't happen in people. But so I'm just going to let you know that. But there is another class of enzymes that you haven't mentioned that this could possibly be, if we're going backwards. Synthetase, and what would we be synthesizing? Succinyl-CoA, and this is succinyl-CoA synthetase. I don't know historically why they named it for the reverse. It's one of those few times, sort of like,
So,
it's just one of those times that it's named for the reverse. It makes sense going backwards. It would be a succinyl-CoA synthetase. I don't know why that. Because it's using GTP for energy. You make succinyl-CoA.
so hypothetically speaking, if we were given something similar to this, it wasn't that exact, those exact molecules, well, how would we go about naming that? Like,
Usually, what I do is I ask you for your rationale.
okay.
To explain it. And so that's why if you had, I mean, there are multiple ways to explain it. And just like we've already seen it once before with, remember how in plants and animals, the carboxykinase peptidase, sometimes they're, depending on who discovered it in the species or the classification system that's used, they do have two different names. Just for that reason. Yeah, no, this is succinyl-CoA synthetase. This is the first time you get energy directly from citric acid cycle. All the other times you made NADH can go on, but we never made something energetic capable.
So, what exactly makes these checkpoints? Because this one's not reversible, but it's not a checkpoint.
It's not. So just because checkpoints are not always due to reversibility. It's due to the fact that they can be allosterically controlled, typically, or be controlled genetically or controlled with one of those methods. Usually what we're talking about is allosterically controlled, or covalently modified. Okay. Many times they are relatively energetically unfavorable going backwards, but not necessarily.
So it's literally like thinking like a checkpoint like a gate where you can shut, close it.
Exactly.
at will. So
This one doesn't. What this would, way to read this is when you make succinyl-CoA, it's going to go on. It's not the determining factor. It's going to go on to make succinate. If it starts to get backed up, the way that it does help control it isn't at this enzyme level. It goes all the way to citric synthase. And citric synthase has a binding site for this, and so therefore it blocks it. Okay. We've actually seen this one before. Okay. The way, because usually we don't utilize GTP specifically for energy. We utilize ATP more often than GTP. And so we have this nucleoside diphosphate kinase. We've seen it multiple times before. And it's a way to get ATP from that GTP. Where literally just the two NTPs, that's why it's got a generic name, just swaps the terminal phosphate. Okay, so that's why sometimes when they draw out the citric acid cycle, it'll look like this. Oh, that's fine. They'll have like the step 0. Ooh. And then they have this little thing here where we have that's probably not the best way. I did it going in the wrong direction, but that's okay. Hopefully you get the idea. GDP, to GTP plus ADP plus ATP. And so then what happens is we get the ATP, but it's not directly a part of the citric acid cycle. It's one step removed. You get the technically GTP and not. And that enzyme right there that I just drew to the side was that nucleoside diphosphate kinase. This is a little FYI. All right. 6 and 7. I don't know why I put them on the same. Okay. I'm not sure why I put them on the same. But once again, I want to give you a chance to try to figure out what's going on, naming them based off of what you know and all that kind of stuff. Now, I don't know if I told you or not. They're not including step 0. There are only 8 steps to citric acid cycle, so we're almost finished. I've forgotten glycolysis to show you some of the videos, or a video or two that people have made in the past. I don't have a good one on citric acid cycle. That's in English. I got one in Spanish. I used to have one in Spanish. It's really fun to hear you speak Spanish. And I think it's still chapter 20 of the book. They talk about FADH2 and making 1.5 ATP. So I think I did update that. We don't cover, due to time constraints, we will not cover the plant chapter. Since almost everybody in here is geared towards biomedical sciences, we just usually don't have time. I'd much rather go further into like looking at the hormones and all that kind of stuff than spending chapter talking about plant biochemistry. Okay, so at the very top on step 6, we have succinate becoming fumarate at the expense of an FAD becoming an FADH2. Please note it's FAD, not NAD. Also, please note FAD is truly FAD, but NAD is NAD+. It's just a little side note. Okay, so what class or classes of enzymes could this be? And it is a dehydrogenase, and if you were going to name it, what would make sense to name it? And that is true. This one is, and I'm going to abbreviate because I don't have that much room. But yes, it's succinate dehydrogenase. Step 7 is an important step. Before I forget, I was about to forget, and I don't know that your book tells you this. Step 6 is the step that is located at the inner membrane of the mitochondria. It's the part that's going to, it's also complex 2 in the electron transport chain. It's the part that's the common junction point between those two metabolic pathways, for lack of a better word. Okay, so step 7, if hydration of fumarate becomes L-malate, and it's fine with me if you just call it malate. That's the L versus the D, just let you know the stereochemistry for the sugar. We've actually seen malate before. We have. I don't know if you remember it or not, but we saw it as part of gluconeogenesis. Malate's important. It's another one that's commonly used in multiple metabolic pathways. This is another one that's a little bit more tricky in naming. Does anyone know what enzyme this one is? It's got a common name. That's the reason why I say it. It makes sense when you hear the common name, but it's Pimarace. Yes. This one's Pimarace. Which I don't know. My Spanish voice kicks in. Pimarace, it makes me think smoking. I have no earthly idea why. I mean, I know why. I have no earthly idea why this is called Pimarace. Or Pimarate. No, it's called Pimarace. if it has anything to do from the common word, because, well, if you speak Spanish, you know what I mean. Fumar, it's to smoke.
Whenever you
Please don't smoke.
step 6 is located in the inner mitochondrial membrane. You also said it was a second point or something.
No, I call it a junction point for the electron transport chain. I don't know that's the best term for it. No, junction point, like in the subway system, you have different lines that have it's kind of like that. It's part of complex 2 of the electron transport chain. Electron transport chain is 4 complexes. There. So this is something that try to keep in your mind. We'll see it again in the future. By the way, it's the reason why you only get 1.5 ATPs instead of 2.5 for FADH2. Okay. Now we've come to the end of our road for the citric acid cycle, at least. This is step 8. We go from malate to oxaloacetate. We get another NADH out of it. We've actually seen this enzyme before. Does anyone remember where did we see this enzyme? I even said, please don't forget. I'm sure, I'm 99% sure that I said that, just like I did for complex 2. Where did we see this enzyme before? You get malate from oxaloacetate.
Getting it out of the mitochondria.
Exactly. This was a part of gluconeogenesis, not directly, because remember, oxaloacetate is a part of gluconeogenesis. It's been a long week, even though it's been a short week. But oxaloacetate do not get to, it can't transverse all the membranes from the mitochondria, right? And so since the gluconeogenesis has to be able to go between the mitochondrial matrix and the cytosol, it utilizes this enzyme. Please note, the way that it's written right here for the citric acid cycle, they show it as being irreversible in a sense, but overall it is reversible because this is how, that's why this is not a checkpoint, because this is how we can effectively get NADH across the membrane as well, because NAD and NADH cannot cross between the mitochondria membranes and the cytosol. So this is one way we can get the electron equivalents. There's a cytosolic form of this enzyme, and then there's the mitochondrial form of this enzyme. What enzyme is this? What class of enzymes can it be? It's a dehydrogenase, and if you're going to name it, you would name it malate or malate dehydrogenase, and that's what it is. Okay. Like I said, it's one that we've seen before. Again, oxaloacetate, maybe you can see it now, you can picture it. It's the alpha keto acid of aspartate. So, if you add everything up, you get per round of the citric acid cycle. So for every 1 pyruvate, you get 1 GTP, which can go on to make 1 ATP. That's why some books call it an ATP. In reality, it's a GTP directly. You get 1 FADH2. You get 4 NADHs. However, one of those came from step 0, this little asterisk. And you kick off 3 carbon dioxides. I wanted to point out the carbon dioxides because I think that we stated this, I don't remember if it was this semester or last semester, because they started blending together. Carbon dioxide is the most oxidized form of carbon. That means you have literally been able to abstract all of the electronic potential, electronic power out of that carbon. Okay. And so it's not good to us anymore. This is one of the reasons why it's a byproduct, a waste product. We want to exhale it out. Yes, there are some enzymes that do carboxylation, but that's minor. Okay, so that's why we used all the energy we could. We used it, kicking it to the curb, so to speak. Does it sound like we're on Springer? Whoops, I did not mean to do that. This is just a little table to show the 8 steps. Please note it does not have, it's from the old book, it doesn't have step 0, pyruvate dehydrogenase complex. A lot of this I've already covered as we went along. The 3 control points or checkpoints, there are 3 within the cycle. Please don't memorize just the numbers, because one tricky thing that I've done before is on exams or quizzes. Instead of having it where step 0, 1's at the top, I turn it, turn the cycle, and when people just memorize the numbers, they get it wrong, okay? So that's why you kind of have to understand the theory, the philosophy, or the reasoning behind it, I should say. But citric synthase, isocitric dehydrogenase, and alpha-ketoglutarate dehydrogenase complex. And then one outside the cycle, which is pyruvate dehydrogenase complex. I've got the word complex on. But if you've got the word complex, it's fine too. I would know what you're talking about. Okay. All right. Are there any questions so far? Citric acid cycle, hopefully by now, it's starting to make a little bit more sense. You're getting a feel for the enzyme. There's the Acetate and Pimarace are the hard ones. The one that's named for it's backwards, but the synthetase, because it uses GTP. I like this little picture here because it just summarizes and shows everything. Shows you the relative locations where stuff occurs. So you can see the overall, the big scheme of things. They don't have the enzyme names in there, but you could even probably use this to kind of quiz yourself on like the enzyme names and functionalities and the fact that where the ATP would go in, or how do you get the ATP, like where's the GTP and all that kind of stuff. Okay. This, I just want to hammer it in. Right? Hopefully this makes sense. If the cell, I'm anthropomorphizing here, is resting, so therefore you do not need any more energy, right? Your ATP tends to be high, ADP would be low, so that ratio is going to be high overall. Your NADH will be high, your NAD plus will be low, so that ratio also will be high. Whereas if your cell is active, the converse is going to be true. And so we are very limited in the actual total amount of the NAD that we have around and the ATP, ADP that we have around. There's only so much, right? So, especially the NAD and ADH, we're really, really limited. And so it's that ratio that's so important. Okay. All right. Oh, this is where we get to, this is the fun part. I get excited. I really like this figure, this kind of thing. They want you to be able, some of you already know, but be able to start to put the pieces together. Okay, this is to show you essentially what we know so far, with the exception that we haven't covered fatty acids yet. But it's showing you like how the pieces start to fit together. Remember that one picture of the metabolic cell? Remember there was all those little pathways and all those junction points, and it just really is overwhelming. Whereas if you look at this, it's just a little bit more simplistic, and so it just has a couple of them. But we've already covered a lot of this. So just to review, the carbohydrates. What's an example of the carbohydrate? Glucose. That's the simplest, but we've also covered things like sucrose and fructose and all that. Those, you break them down, you get pyruvate using pyruvate dehydrogenase complex will go on to make acetyl-CoA. Acetyl-CoA then is utilized to make a lot of these different metabolic intermediates. Okay. Likewise, I've already told you the breakdown of fatty acids. We're going to find out later on. It makes acetyl-CoA to enter in the citric acid cycle. We've talked about proteins. What class of enzymes break proteins down to amino acids? Proteases. Okay, you break it down to your specific amino acids, and we're going to find out, we actually already know some of these, but these amino acids can then also be catabolized to get energy. Okay, so you can use it. The amino acids are the only, or proteins are the only one of those classes that we can't store for long-term storage, right? You can store some glycogen, carbohydrates. You definitely store a lot of store, myself included, way too much fat, right? The fatty acids for energy. But we don't store proteins. There's no long-term storage for that. In particular, yes, you can build up muscle, but that's one of the reasons why your muscles can decrease relatively quickly as well, because of the fact that it will catabolize that amino acids. So for an example, then we break down the proteins, we get alanine. Alanine can be converted to pyruvate using what enzyme? Alanine, aminotransferase. Okay. Then we could have aspartate. Aspartate can break down and turn into what? which of these? Aspartate.
Oxaloacetate.
Oxaloacetate, and what enzyme, what specific enzyme makes that, that does that? So think about, if it's alanine aminotransferase that goes from alanine to pyruvate, what aminotransferase is going to go from aspartate to oxaloacetate? Aspartate aminotransferase, okay? We could have, and we'll find out asparagine actually can also go that way. Asparagine can become aspartate, aspartate can become, right? So we have glutamate, we've already covered glutamate, can become which of these? Alpha ketoglutarate. What class of enzymes can do it? It's an aminotransferase. This is a little asterisk. Keep this in your mind, you'll probably forget it. All aminotransferases do that. Alpha ketoglutarate and glutamate are involved in every single aminotransferase. Okay, so there's not one called glutamate aminotransferase. It's a little side note. Okay. We'll cover that whenever we start talking about nitrogen. How do you get rid of the nitrogen and glutamate? It's kind of a stickler. What? Well, it's hard. Okay, and then we'll cover this. So that's why we can see how it's used in catabolism. So if you prefer seeing a cartoon look, here we go. Give me a moment to take in the beauty of the mitochondria of God's creation. Here. Some of this we already know. We've already covered. Some of it we will cover throughout the semester.
So this is not to scale, right?
This is not to scale, no. All right. Does anyone want to hypothesize, knowing what you know now? So we have this alanine becomes pyruvate. So where they put a TA, what is that? It's aminotransferase. The old name, or like a common name for aminotransferases, were called transaminases, which sounds like a hot rod car. But so that's why that means it's TA for transaminase. It's transaminase reaction. But it's aminotransferase, and it's alanine aminotransferase. Okay. What we'll find out, oh, well, this is just glycolysis. We've already done that one. We won't cover all of these. We cover some of them specifically. And as we mentioned here, asparagine becomes aspartate, aspartate becomes oxaloacetate, oxaloacetate becomes malate, malate can cross over, and then we just get the reverse, and that's how you can get it through citric acid cycle. Over here, we have some of the more complicated ones become acetyl-CoA, and then transfer into, and so on and so forth. Okay, glutamine, I don't know why they did this, but glutamine can become glutamate, glutamate can become alpha-ketoglutarate, which goes on in as well. Okay? All right. Here we actually already get the answers. So transamination is the old name. It sounds really cool though. It's aminotransferase. And once again, specifically, this one would be aspartate aminotransferase. This one is just aminotransferase. It'll make more sense once we get to it. Because your aminotransferases are reversible, so at all points in time, you have to have something that accepts the amino group. So you can imagine if you're breaking aspartate down to get oxaloacetate, the nitrogen that it's being transferred to is alpha-ketoglutarate, so you make glutamate. Whereas, let's say that we want to go from oxaloacetate to aspartate, what happens is oxaloacetate takes a nitrogen from glutamate, so that way you make aspartate and you get alpha-ketoglutarate out of it. Same thing with alanine. So this alpha-ketoglutarate and glutamate is always they're called ping pong reactions, so they go back and forth, and it's always one of the partners, one set of partners, in the doubles reaction. Yep.
So if you add more oxygen, it would go more to the ketoglutarate?
No, it's not just based off of the oxygen. They're showing it's reversible. It's just to show you some examples. It's going to depend on lots of different things. If you need, let's say that we need aspartate, and this is the way that we're going to make it, then we're going to take it away from glutamate to make aspartate. If you need glutamate, one way to make glutamate is to get it from alpha-ketoglutarate and we can literally take the nitrogen from any of the other amino acids. Okay. So trust me, we go over lots of examples of it throughout the semester. We see aminotransferases over and over and over and over again. You have to be able to replace the intermediates, if you take it away, and that's where some of the suggested problems are similar to that, to where, and there are a lot of medical conditions associated with this, that you can imagine that there are ways, and sometimes they're called shunts, ways to get around parts of it, because if you take something out of the citric acid cycle, you've got to be able to replace it. As we've already seen, there are multiple ways to feed back into it. Okay, and so this is just one of the ways. This is pyruvate carboxylase. Just to show you from before. Okay. So that was another example for anabolism. Remember, catabolism is breaking down, anabolism is building it up. This one is a little cartoony way, or one of the cartoony ways of showing that same thing. Okay. Please note, this isn't my favorite one because it doesn't show you, it shows you things that we can't do. Okay. And so that's why we don't go this way, we go this way, but we, as in mammals, this is the way that we go for anabolism, but there are other animals or I shouldn't say animals, there are other organisms that can go about different ways as well, okay? But this is just to show you that we can go ahead and feed back into to citric acid cycle. And this is the not drawn to scale mitochondria one. Okay. Which should look really familiar in the sense that essentially it's gluconeogenesis. We saw this, we just added in the circle part of the citric acid cycle. Show how the citric acid cycle can actually help with gluconeogenesis. If we don't need the energy, then we can build it up going backwards to store the sugar. Are there any questions so far? Awesome. All right. So we've tied it together to proteins and we've tied it together to carbohydrates. This is how it's also involved with lipids. So lipids in particular, we're talking about acetyl-CoA. Acetyl-CoA and lipid anabolism is in the cytosol. So and you may want to do this, like you know how I say you have like a page where you're putting in all your enzyme names, enzyme family names, which I suggest that you do that. You may also have one where a cell that you start to put in, like where does certain things go? We had glycolysis occurring in the cytosol. We had citric acid cycle starting primarily in the matrix, with the exception of the one little crossover into the mitochondrial inner membrane. We have gluconeogenesis, which goes through both. But we have lipid anabolism, which we will cover in further detail later on. But it's in the cytosol. Okay. Which does anyone know where this lipid? Where's the opposite of lipid anabolism? So which is beta oxidation. Where does that occur? It's not in cytosol. It's one way that we can keep it compartmentalized. And so that's just it. Keep in mind. Okay. So we have to have some way to get around it. Oh, you can ignore this one. I've already fixed it. I forgot to take that point out. Oh, it's because now it's a PDF. That's why I didn't do that. Okay. Because originally it had a typo in it. And it was a picture, so I couldn't go in and fix it myself. But I found it right. Okay. So acetyl-CoA is just like what we've been talking about with NADH and oxaloacetate. It has problems sometimes getting from one place to the other. And so we have to have an indirect transfer to do that. And so this is one way to do this. And on your own you can go through. But it utilizes citrate. Okay. We'll go into more detail later in the semester when we go over lipids. I just wanted to show you that it wasn't limited only to proteins and carbohydrates. So this is how, and this is also another way that you can effectively get an NADPH. Remember we also talked about the pentose phosphate pathway. This is like a backup to get ADPH. So, yeah, no, so I don't know if there's anything else that you wanted to talk about. Are there any questions? Because I got a little cartoon to show all this at this moment. And then you see it word-wise first. Okay. So here is the not drawn to scale mitochondria depicting this. There's a lot going on, but it's letting you know like how the anabolism is working and how we can build other things from it as well. Some of the cool things I just want to point out. I mean, we've already seen this in gluconeogenesis. We've already talked about, to make proteins with various amino acids. Okay, but I want to point out this is one of the ways where we can go ahead and make the fatty acid derived stuff. But we can also make porphyrins right here. Okay. And one of our porphyrins, we cover this later on. You can almost turn porphyrin ring into like a devotional. What are the porphyrins? We've seen it once or twice so far, primarily last semester actually. What's a porphyrin ring? What's it in or make up? One of the diseases, there's lots of diseases associated with it. One is called porphyria. Some people equate to vampirism.
Is it similar to an aquaportin?
No. Porphyrins are make up the heme ring. Hemes for hemoglobin and other cytochromes and stuff like that. And so, which are huge. These are these huge, relatively planar looking molecules. They are made up from really, really tiny things. And so the amino acid that's the building block for it is glycine. The simplest amino acid can actually be used to build one of the most complex things. And so that's why I say you can actually convert that really into a devotional almost. Okay. I've got one or two slides that talk a little bit about plants, but then the medical implication of this, I want to say, because I like to go over some of the medical diseases associated with the citric acid cycle in greater detail. So I want to save that for Monday. And so are there any questions? No, we only have a couple slides left. Are there any questions? That way we can go into, spend more time talking about why some of these diseases, I don't want to say make sense, but why they make sense logically, metabolically. Any questions? Okay.